Hello everyone, today we will discuss the peroxisomes and its structure. These are the cells, tiny detox and recycling crew. First, we should take a glance over what are peroxisomes and their function in brief. As the peroxisomes are the membrane bound organelle, these are found in eukaryotic cells and they have a different key roles in metabolic processes, which includes the breakdown of fatty acids, detoxification of ROS means reactive oxygen species, and they synthesize the certain lipids which are of main significant. They are characterized by their capability to use molecular oxygen to remove hydrogen atoms from organic substances, producing hydrogen peroxide as a byproduct during the metabolic activities. And hydrogen peroxide is a highly reactive toxic components which are later utilized in oxidative reactions. So in detail we will know the intro step by step later. First we will understand the structure of peroxisome. This is the structure of peroxisomes. Peroxisomes main function is to break down the very long chain fatty acids, branched chain fatty acids, de novo lipogenesis and bile acid synthesis intermediates. So step by step we know the how very long chain fatty acids are broken down, how the bile acid synthesis occurs, how the branched chain fatty acids are broken down, and what is the role of this crystalline core of oxidases. So this is the 3D structure of peroxisomes. First of all, we will try to know the crystalline core of oxidase. This one is the innermost structure which represent the dense crystalline core. This core is packed with high concentration with high of enzymes, particularly oxidases. So in one line we can say that this is the high concentration zone consisting of enzyme oxidase. This is an important enzyme, that is oxidase. Now we will take an example through which we try to know the role of oxidase. For example, in many animals, this core is made up of urate oxidase, which break down uric acid. While human lacks this specific enzyme, the core concept of a dense enzymatic center remains. These oxidases generate H2O2 means hydrogen peroxide as a byproduct, which is a reactive molecule. Hydrogen peroxide is a very reactive molecule that gives the peroxisome its name. Means the concept is that the name of peroxisome is derived from the H2O2 means from hydrogen peroxide the name of peroxisome is delivered. The peroxisome then uses another enzyme, that is catalase. The peroxisome uses another enzyme, that is catalase, which safely convert H2O2 into water and oxygen. Catalase is the enzyme which convert H2O2 means hydrogen peroxide into H2O means water and oxygen. So the motive is that the peroxisome has a good and specific enzyme, that is catalase 1, which safely convert H2O2 means highly toxic molecule into the water and oxygen. Now next is the membrane and transporters. This is yellow line shows the peroxisome is enclosed by a single membrane This yellow membrane shows the structure that peroxisome is enclosed inside this single membrane, which contains various transporters, proteins that control enters and exit the organelles. Means these are the membranes and transporters. So this yellow line is the membrane in which peroxisomes are enclosed, and green line is the transporters of protein that controls the enters, means what type of molecule should be entered and what types of molecule should be exit. So this is the main function of membrane and transporter proteins. Next is the very long chain fatty acid or very long chain fatty acyl coenzyme A. Peroxisomes are the primary site for initial breakdown, means beta oxidation, for very long chain fatty acids. So through the process of beta oxidation, very long chain fatty acids are broken down. There are more than 22 carbon atoms are present in fatty acids. For example, if peroxisomes are dysfunctional, means suppose peroxisomes are dysfunctional, then what will happen? Then certain types of genetic disorder will be induced in form of X-linked adrenoleukodystrophy. Means in one line we can say that very long chain fatty acids are broken down by the peroxisomes. There are 22 carbon atoms in fatty acids. So suppose if peroxisomes are dysfunctional, then what will happen? So the result will be that very long chain fatty acids will accumulate in the body. Consequences will be occur in form of genetic disorder in form of XALD, means X-linked adrenoleukodystrophy, which damage myelin sheath of nerve cells, leading to severe neurological problems. So we can understand this if very long chain fatty acids are not broken down by the peroxisomes, because peroxisomes are not doing well or proper functional. If they becomes dysfunctional, genetic disorder will be occur in form of X-linked diseases, that is XALD, means X adrenoleukodystrophy, which damage the myelin sheath of nerve cells. Resultant will be occur in form of severe neurological problems or disorders. Next is the branched chain fatty acyl coenzyme A. Peroxisomes are essential for breaking down branched chain fatty acids which cannot be metabolized into the mitochondria. The story is that why peroxisomes are important, because branched chain fatty acids are not metabolized into mitochondria. So there is an importance of peroxisomes. Peroxisomes broken down these branched chain fatty acids. For example, phytanic acid, it is a branched chain fatty acids found in dairy products. This is found in dairy products, animal fats and some fishes. The inability of breakdown phytanic acid due to peroxisomal defect leads to Refum disease, causing symptoms like nerve damage, vision loss, and skin problems. So, in one line, we can say that if branched chain fatty acids are not metabolized into the mitochondria, so the peroxisomes will broken down the phytanic acid, means branched chain fatty acids, into smaller ones. If peroxisomes dysfunctional, then the broken down of phytanic acid will not be occur. It is mainly found in dairy product, animal fats, and some fishes. So, if broken down of phytanic acid will not be occur, the diseases will caused in form of Refum disease, which will affect the nerve cells, vision loss, and skin problems. Next is the bile acid synthesis and intermediates. Peroxisomes performs the final steps in the synthesis of bile acids from cholesterol. For example, the synthesis of primary bile acids into the cholic acid and chenodeoxycholic acid and requires peroxisomal enzymes. These bile acids are essential for digesting fats and absorbing fat soluble vitamin in the intestine. Next is the de novo lipogenesis. De novo means from scratch and refers to the peroxisomes' role in certain lipids type creation. For example, peroxisomes are vital for synthesizing plasmalogens. Plasmalogens is a special class of phospholipids that are critical component of cell membranes, especially in brain and heart tissues. A deficiency of plasmalogens can lead to severe developmental and neurological disorder. So, in whole, we can say that the component of peroxisomes, these are very long chain fatty acid, branch chain fatty acids, bile acid synthesis, lipogenesis. If peroxisomes will not work properly, these will accumulate inside the body and induces many types of neurological problems and disorders. Next is the introduction to peroxisomes. In detail, we will know the role of peroxisomes, how they work, from where they originate, and what is their specific role in our body. So, what are peroxisomes? First, we should know what are peroxisomes. Peroxisomes are the tiny membrane enclosed organelles found within the cytoplasm of most eukaryotic cell, including those of animals, plants, and fungi. Essential for cellular health and homeostasis. How? These are indispensable for maintaining cellular health due to their central involvement in several vital processes, including detoxification, lipid metabolism, and energy regulation. We will know these of three step by step in detail. Detoxification, how peroxisome detoxify the waste material or the waste components. So, a primary and highly critical function of peroxisome is the neutralizing of hazardous substances, most notably hydrogen peroxide H2O2, which is a toxic byproduct generated during normal metabolic activity within the cell. Without functional peroxisomes, cell would accumulate severe oxidative damage, which can lead to cell dysfunction and contribute to the development of various diseases. This oxidative damage can harm to the DNA, proteins, and cell membrane, accelerating cellular aging and increasing the risk of numerous diseases. So, in this format, these detoxify the hazardous waste and protect the cellular components. Peroxisomes are characterized by their component of multiple oxidative damage that performs crucial metabolic reactions. These enzymes enable peroxisomes to broken down. So next we will know primary mission of peroxisomes. These of we already known tiny membrane bound organelles essential for cellular health and homeostasis. Core functions detoxification, as I already mentioned, lipid metabolism, and energy regulation. So in this way, these enzymes enable peroxisomes to break down fatty acids, synthesize specific vital lipids, and shield the cell from toxic molecules. Furthermore, peroxisomes play a fundamental role in cellular homeostasis through their participation in bile acid synthesis, cholesterol regulation, and the maintenance of energy balance. So, in this way, we are trying to know that what are the peroxisome and how do they work. Next is the structure and functionality of peroxisomes. If we talk about their size, means appearance, so peroxisomes typically appear as a small spherical organelles. They are encased by a single phospholipid membrane, often described as a lipid bilayer membrane. In terms of size, peroxisomes are comparable to lysosome, generally ranging from 0.1 to 1.0 micrometers. This makes them tiny, roughly 100 to 1000 times smaller than the width of a human hair. So this is about the size. Key distinction means a significant distinction that sets peroxisomes apart from other organelles like mitochondria and chloroplast is their lack of DNA. Consequently, all their necessary proteins are synthesized in the cell cytoplasm and must then be imported into the peroxisome. These proteins are encoded by nuclear DNA, transcribed into RNA, and then translated into. So, these proteins are encoded by nuclear DNA and transcribed into RNA and then translated into protein by cytoplasmic ribosomes. So, the architecture of a peroxisomes is defined by several key components, that is lipid bilayer membrane, catalases and oxidases, and transport of proteins. So these of key component we will know step by step in detail. So first of all, lipid bilayer membrane. This outer boundary act as a protective fence, means this act as a protective fence. It meticulously regulates the transport of enzymes and metabolites into and out of the organelle. Crucially, it is also function as a containment field, preventing the toxic byproduct generated inside, such as hydrogen peroxide, from leaking into the border cytoplasm and causing widespread cellular damage. This membrane is studded with special doorways or transport proteins. So these green act as a transport protein, transporters. Transporter proteins act as gatekeepers, actively pulling specific protein made into the cytoplasm into the peroxisomes. It also contains pumps that facilitate the movement of other molecules. So this is about the lipid bilayer membrane. Next is the catalases and oxidases. Catalases in peroxisomal matrix, the semi-fluid enzyme-rich compartment inside the membrane is packed with powerful enzyme, that is catalases. Catalases is the primary and most famous enzyme which is responsible for efficiently breaking down H2O2. So catalases is for breaking down H2O2, means hydrogen peroxide, into harmless water and oxygen, acting as a rapid demolition expert to prevent cellular damage. A single catalase enzyme can catalyze the decomposition of millions of hydrogen peroxide molecules per second. Other oxidases aid in breaking down long chain fatty acids and detoxifying harmful substances. These oxidative enzymes initiate reaction by removing hydrogen atoms from molecules, consuming oxygen and producing H2O2 as a byproduct in some animals, particularly in liver and kidney cells. A dense, this is a crystal like core which I am talking about, can be observed into the matrix, which is a tightly packed cluster of enzymes like urate oxidase involved in breaking down waste product. Next is the transport proteins. So transport proteins, these are the transport proteins. As mentioned, these proteins are embedded within the membrane, facilitate the import of newly created enzymes from the cytoplasm into the peroxisomes, from cytoplasm to peroxisomes, and assist in efficient removal of metabolic waste products. So we talked about lipid bilayer, catalase and oxidases, and transport proteins. Next is the adaptive nature, in which we will talk about peroxisomes just not exist, they respond to the cell's need, replication, adaptability. With an example. So, unlike many other organelles, peroxisomes replicate through a process of growth and binary fission. Growth and binary fission. This means they can grow and divide independently within the cell. This dynamic capability ensures that the cell can rapidly adjust its peroxisome population to meet fluctuating metabolic demands and detoxification challenges. Detoxification challenges means how many types of metabolic byproduct are producing inside the body that may be toxic to the body. So there is essential for detoxification processes. Detoxification challenges. For example, if we consume a lot of fatty food, our liver cells may sprout more peroxisomes with packed fat digesting enzyme to handle the metabolic load. So this is the adaptive nature of peroxisomes. Next is the role in detoxification, means the role of peroxisome in detoxification, how they detoxify the byproduct which are made during the metabolic processes, in which we will know breaking down of hydrogen peroxide. Cells are the primary site for neutralizing cellular toxins. Breaking down of hydrogen peroxide, eliminating harmful compounds like alcohol, drugs, phenols, and neutralizing reactive oxygen species. The most important feature is how reactive oxygen species are formed and how they are neutralized by the enzymes. So we will know it step by step. First, we will know, so a key and primary function of peroxisome is the neutralization of toxic substances. Neutralization of toxic substances that compromise cellular integrity. Metabolic processes continuously generate ROS, means reactive oxygen species. This is very important to know what are these reactive oxygen species. So during the metabolic processes, ROS means reactive oxygen species are formed continuously, such as hydrogen peroxide, H2O2, as byproduct. ROS can inflict severe damage to DNA, proteins, and cell membranes. Severe damage, thereby accelerating cellular aging and disease progression. So this is about the how peroxisomes work in detoxification. Now we will know step by step. Breaking down of hydrogen peroxide occurs, in which format a peroxisome broken down the hydrogen peroxide. So this is breaking down hydrogen peroxide is a natural byproduct of fatty acid oxidation and other metabolic reaction. The peroxisomal enzyme catalase protects cellular components by breaking down toxic H2O2 into water and oxygen. For example, liver generate H2O2 by breaking down alcohol and drugs. Without peroxisomes, toxic H2O2 would accumulate, leading to liver damage. So this is about how hydrogen peroxide is broken down by the enzyme catalase. Harmless component, water and oxygen. So this is about the broken down of toxic product of H2O2. Next, we will know the eliminating harmful compounds. So in eliminating harmful compounds, beyond hydrogen peroxide, peroxisomes adapt at detoxifying a wide array of harmful substances, including alcohol, means ethanol, drugs, formaldehyde, a metabolic byproduct, methanol, phenols from plant and pollution, and nitrites, which can be found in food and the environment. This activity is particularly pronounced in the cells of the kidney and liver. These are more pronounced in the cells of kidney and liver, which serves as the body's primary filtration system. Approximately 1 to 2% of the cell mass in liver is composed of peroxisomes, reflecting their intense detoxification activities in these organs. In these means kidney and liver organs. So this is how eliminating harmful compounds are detoxify in liver and kidney by breaking down alcohol, drugs and phenols, ethanol, formaldehyde and nitrites. Next, we will know neutralizing reactive oxygen species by actively scavenging excess ROS. Peroxisome play a vital role in preventing oxidative stress and subsequent cell damage. This detoxifying cell is especially important in energy-intensive cells such as neurons, which are highly susceptible to oxidative damage due to their high energy requirements. By shielding these nerve cells from oxidative damage, peroxisome help reduce the risk of neurodegenerative diseases like Alzheimer's and Parkinson's. So, this is the role of peroxisome in neutralizing the ROS, which is very harmful to the brain and its cells. So, in this way, peroxisome provides a shield to the nerve cells and keeps them healthy. Now, next is the role of peroxisomes in lipids, metabolism, and energy. In this, we will know that how they manage the fat, energy and many more. So first we will take a look on peroxisoma's central hub for lipid processing. How? In addition to their role, detoxification duties, peroxisomes are the major player of lipid metabolism. Their functions, which include breaking down fatty acids, synthesis of bile acids such as regulation of cholesterol, are essential for sustaining cellular energy levels and preserving this membrane structure. It is very important to keep this structure preserved because this structure decides what should be out and what should be inside the organism. So previously we will know about the peroxisomes, a central hub. Now we will know the key metabolic roles of peroxisomes in beta oxidation of fatty acids. In this, peroxisomes are equipped with breakdown long chain fatty acid, breakdown of very long chain fatty acids, breakdown into smaller molecules. These smaller molecules such as acetyl coenzyme A transported to mitochondria for complete oxidation and energy generation, and energy generation. Critically, peroxisomes are uniquely capable of processing very long chain fatty acids which have 22 or more carbon atoms and are too large for mitochondria to handle directly. They also break down branched chain fatty acids like phytanic acid, which cannot be metabolized into mitochondria. For example, in muscles and liver cells, during period of fasting, peroxisome initiates the breakdown of very long chain fatty acids to provide vital energy source. So this is how beta oxidation of fatty acids occurs. They are shortened very long chain fatty acids that are too big for mitochondria to handle directly, and peroxisome preparing them for energy production. In this manner, they broken down into the smaller molecules and then they are transported to the mitochondria for further process. Next is the bile acid synthesis. In bile acid synthesis, the creation of bile acids which are necessary for digesting fats in the intestine is heavily dependent on peroxisomal enzymes. In liver cells, peroxisomes modified cholesterol to produce these essential acids such as cholic acid and chenodeoxycholic acid, thereby facilitating the digesting and absorption of dietary fats and fat-soluble vitamins. If any defect occurs in this process, it can lead to the accumulation of toxic intermediate, disrupting fat absorption, causing liver injury, as seen in Zellweger syndrome. So this is about how bile acid synthesis works and convert cholesterol into bile acids which are essential for digesting dietary fats. Next is the plasmalogen synthesis. So now we will talk about plasmalogen synthesis. Peroxisomes primary site for synthesis of plasmalogens. A specialized class of phospholipid. These molecules are absolutely important for the structure and function of brain and nerve cells. So the plasmalogens are the special class of phospholipids which are absolutely important for the structure and function of brain and nerve cell membranes, structurally reinforcing myelin and neural membranes to enable rapid nerve signaling. Without enough plasmalogens means if there will be no enough plasmalogens, the consequences will be in form of they will not work properly. If any deficiency in plasmalogens is linked to severe neurodevelopmental disorders and cardiac diseases. Next is the purine processing. Now we focused over purine processes. In this, peroxisomes helps break down purines. Purines are adenine and guanine, which are building block of DNA and RNA. One enzyme converts a purine breakdown product into uric acid, which is then excreted from the body. This process needs to be balanced as too much uric acid can cause problems like gut. So this is the purine processing help break down the building block of DNA and RNA. Next is the peroxisomal disorders. In this, we will know the what types of disorders can be occurred if peroxisomes works not proper. Malfunctions in peroxisomes biogenesis or enzyme function can lead to a group of severe genetic conditions known as peroxisomal disorders, often resulting in profound metabolic dysfunction and neurological impairment. These disorders starkly illustrate the non-negotiable role that peroxisome play a human health and proper development. Some well-known peroxisomal disorders include Zellweger syndrome, Adrenoleukodystrophy, and rhizomelic chondrodysplasia punctata disorder. These are the three disorders which we have to discuss. So, in this means Zellweger syndrome, this is a rare and devastating genetic condition characterized by the complete absence or dysfunction of functional peroxisome. It leads to profound brain damage, severe brain malformation, and liver dysfunction, often accompanied by significant developmental delays and skeletal abnormalities. Infants diagnosed with this syndrome face an extremely poor prognosis, with most stumbling before their first birthday. This disorder is a severe consequence of a failure in protein transport into the peroxisome. Next is the adrenoleukodystrophy syndrome. This genetic syndrome occurs when peroxisome fails to break down very long chain fatty acid. Means adrenoleukodystrophy is the outcome of that the very long chain fatty acids are failed to convert into smaller ones because they will not broken down and they accumulate inside the body, and these are toxic to the myelin sheath of nerve cells, leading to progressive demyelination, severe neurological decline, and muscles weakness. The well-known film Lorenzo's Oil is based on the true story of a family searching for a treatment of their son with ALD. Next is the rhizomelic chondrodysplasia punctata RCDP. It is a disorder which arises. So we are discussing this one. Rhizomelic chondrodysplasia punctata RCDP. This is disorder which is arise from the specific defect in peroxisomal synthesis of plasma logens defects. It is characterized by several skeletal abnormalities, profound intellectual disabilities, and the development of cataracts. Now next is the peroxisomes in different organisms. Different organism means in animals, in plants, in fungi and protists. A universal organelle with specialized job. Peroxisomes are a common feature of eukaryotic cell. Their specific function can be adapted to the needs of the organism. In animal cells, peroxisomes play a key role in detoxification, fatty acid degradation, and bile acid synthesis. These organelles are particularly abundant in liver and kidney cells. This particular organelles means peroxisomes are abundant in kidney and liver cells, where their function are essential for maintaining cellular health and handling intense detoxification activities. In plant cells, peroxisomes in plants are sometimes called glyoxysomes in plants. They are important for photorespiration, a process where plants recycle toxic byproducts of photosynthesis. Important for photorespiration, a process where plants recycle toxic byproducts of photosynthesis. They also play a vital role in germination of seeds by converting stored fatty acids into sugar, providing the necessary energy for the young plant to grow. In fungi and protists, in these organisms, peroxisome assist in metabolizing complex organic molecules to generate energy for cellular functions. For example, peroxisomes in certain yeast species are involved in metabolic pathway that ferment sugar into alcohol ferment. Next is the peroxisomes in aging and medicine. In this, we will know the role of peroxisomes in aging and medicine. In aging, research indicates that as cells age, the efficiency of peroxisomal function tends to decline. This reduction can lead to an increase in oxidative stress and a higher susceptibility to age-related metabolic disorders. Consequently, scientists are actively exploring therapeutic strategies to enhance peroxisome activity to promote healthy aging and combat diseases. Therapeutic potential in this peroxisomes and longevity. Evidence suggests this boosting peroxisome function can improve the health of brain, muscles, and metabolic system. Certain dietary choices such as consuming omega 3 fatty acids have been shown to support peroxisomal efficiency. Peroxisome in cancer research. An emerging area of study is the role of peroxisome in cancer. Some types of cancer cells appear to suppress peroxisomal function to better survive in their own toxic self-created microenvironment. This has led to investigations into peroxisome activating drug as a potential strategy to selectively target and eliminating cancer cells. So this is about the cancer research, how the peroxisome works to eliminate the cancer cells. Next is the protein transport into peroxisomes. Protein transport means targeting into peroxisome. As peroxisome their own DNA and ribosome lack their own DNA and ribosome. All their necessary proteins are synthesized in the cytoplasm and must be imported. To send any protein from cytoplasm to peroxisome, a special signal is required known as peroxisome targeting sequence. In short, we can say PTS. They are of two types, PTS1 and PTS2. In PTS1, C-terminal signal. So PTS refers to the peroxisome targeting sequence, means from cytoplasm to peroxisome, how the process occurs. So this adapt peroxisome targeting sequence, this is known as PTS. Protein needs a shipping level that is known as peroxisome targeting sequence. In short, it is known as PTS. And it is of two types, PTS1 and PTS2. In PTS1, this is C-terminal signal. In this is located at the C-terminal end of the protein. Structure is a specific sequence of just 3 amino acid sequence, that is 3 amino acid, serine, lysine and leucine or SKL at the end of the protein. This is most common one. Prevalence, the majority of peroxisomal protein utilize this PTS1 signal for import. Second is the PTS2 and terminal signal location. This is the location. It is at the N-terminal end of the protein. Structure is longer and more complex sequence, typically consisting of 9 amino acids. This is the basic difference between the PTS1 and PTS2. There is 3 amino acid and there is 9 amino acid sequence. The process of protein transport for lumen protein by a PTS1 pathway is a multi-step mechanism. A step-by-step guide to protein import. How the protein import to peroxisomes. The PTS1 pathway, import pathway. In this recognition, in the cytoplasm, a soluble receptor called PAX5 recognizes and binds specifically to the protein's SKL sequence. The protein can be in its fully folded state when it binds to the PAX5. Now the docking. PEX5 protein travel to the peroxisomal membrane and binds to the PEX14, which is located on the membrane. Next is the translocation following docking. The complex is transferred to the translocator channel. This channel is composed of a complex protein, specifically PEX2, PEX10 and PEX12, using ATP for energy. This channel opens and the protein is transported into the peroxisome's interior known as lumen. So these enters the lumen. Next is the recycling. The PEX5 is tagged and sent back to the cytoplasm to be reused. How? Once the protein has been successfully delivered into the peroxisome, it is important to PEX5 to send it back to the cytoplasm to facilitate the import of more proteins. This recycling process involves PEX4 adding a ubiquitin tag to PEX5 and then PEX1 and PEX6 utilize ATP to release PEX5 from the membrane for its return to the cytoplasm. So this is the process. The transport of protein designed for the peroxisomal membrane, PMP peroxisome membrane protein, follows a slightly different process. In the cytoplasm, PEX19 acts as a soluble receptor. On the peroxisomal membrane, PEX3 and PEX16 serve as the docking sites for these membrane proteins. Now next is the peroxisomes formation and fission. In this, we will know how peroxisomes multiply. The cell increases its peroxisome population and metabolic demand in which way? A two-step process. So first, peroxisomes have the remarkable ability to increase their number through a process called fission. Means peroxisome can increase their number as per demand by the process of fission. This ensures the cell can adapt its peroxisome population to its metabolic needs. Next is the how protein import. This one. The formation of new or small peroxisome begins with the insertion of protein. First, membrane protein are inserted, followed by the lumen proteins like catalase, using the protein transport processes described later. Next is the fission. Once a peroxisome grows larger, the PEX11 protein play an important role. PEX11 constrict the peroxisomal membrane, effectively dividing into two smaller peroxisomes. Cells that require more peroxisomes, such as those with high metabolic demands, often exhibit higher level of PEX11, facilitating this increase in peroxisome number. Lastly, in summary, we will know the brief of the peroxisomes functions from top to bottom. In summary, peroxisomes are profoundly important organelles for detoxification, lipid metabolism, and overall maintenance of cellular homeostasis. They actively prevent oxidative damage. This is the important feature. Prevent oxidative damage by breaking down harmful substances like H2O2, catabolize toxins, and contribute significantly to cellular energy production through the fatty acid breakdown. The severe consequences of peroxisomal defects, including devastating genetic diseases like Zellweger syndrome, Adrenoleukodystrophy, and rhizomelic chondrodysplasia punctata, underscore their critical importance to human health and development. From neutralizing toxic chemical in the liver to support complex brain functions through plasma login synthesis, peroxisomes performs an array of tasks that are vital for cellular and organism health. Ongoing research into their function holds significant promise for developing novel treatment for metabolic diseases, neurodegeneration, and even cancer, positioning peroxisomes as a key area of focus in modern biomedical science. Now, lastly, we will take an example through which we will understand the peroxisome in few words. Imagine your body's cells as a bustling city, always striving for efficiency and safety, while the mitochondria are the grand power plant generating energy, and the nucleus is the city hall managing blueprints. Peroxisomes are the city's unsung specialized recycling and hazardous waste management crew. They operate in tiny self contained bubbles, diligently breaking down the toxic byproduct of the city's metabolism and refining complex fuels into usable energy components, all whilst also building important infrastructure materials like plasmalogens for vital communication networks. Without these diligent microscopic crews, our cellular cities would quickly succumb to pollution and energy crisis, leading to widespread dysfunction and eventual collapse. If you have any questions regarding this topic, please leave them in the comments section. If you like this video helpful, like, subscribe and ring the bell so never miss out. Thanks for watching.